Imagine trying to listen to a crucial message in a room full of chatter and distractions. That is very similar to the challenge neuroscientists face when attempting to understand the intricate electrical and chemical language of the brain. Signal-to-noise ratio, or SNR, in neuroscience is a crucial measure that quantifies how much meaningful electrical or chemical activity from the brain stands out against unwanted background interference. Essentially, it tells us the strength of the desired signal relative to the level of undesired noise, with a higher SNR indicating clearer and more interpretable data. The fundamental principle behind SNR is to distinguish genuine neural events from everything else. The signal can be anything from the precise firing of individual neurons to macroscopic field potentials recorded from many cells. Noise, on the other hand, comprises any activity not directly related to the brain process being studied. This includes biological artifacts like muscle activity, eye movements, or heartbeats, as well as environmental interference from power lines or electronic devices. Additionally, there is always intrinsic noise within neural systems themselves, stemming from the random nature of ion channel openings and neuronal excitability. A good SNR ensures that observed brain patterns are truly representative of neural function and not just random fluctuations. For researchers and practitioners, understanding and optimizing SNR is critical for designing effective experiments and interpreting data accurately. This knowledge directly influences choices in experimental setup, such as using shielded rooms to minimize environmental interference or selecting appropriate electrodes and amplifiers with low inherent noise. Furthermore, SNR guides the application of various data processing techniques, including filtering to remove specific frequency bands of noise, and powerful methods like signal averaging to enhance consistent brain responses while random noise cancels itself out over multiple trials. Delving a little deeper, we consider both extrinsic noise, which originates from external sources, and intrinsic noise, which is inherent to the biological system. Techniques like adaptive filtering can dynamically adjust to noise characteristics, improving signal extraction even in challenging environments. Another powerful tool is ensemble averaging, where repeated measurements of an event are averaged together. Because the true signal is consistent across trials while noise is typically random, averaging many trials causes the random noise to diminish, effectively boosting the SNR of the underlying brain activity. Ultimately, a robust signal-to-noise ratio is foundational for making reliable scientific discoveries and developing effective neurotechnologies. It allows us to accurately map brain function, decipher neural codes, and understand the mechanisms underlying cognition and behavior. By prioritizing high SNR, we ensure that our insights into the brain are precise, robust, and truly reflective of its complex and fascinating operations.